The nuclear envelope is a crucial structure in eukaryotic cells. It forms a double membrane barrier that separates the nucleus from the cytoplasm, protecting the cell's genetic material while maintaining distinct cellular compartments. The nuclear envelope is a double-layered membrane structure that completely surrounds the nucleus. This barrier separates genetic material from the cytoplasm, creating two distinct environments within the cell. Think of it as a protective wall with controlled entry points. The nuclear envelope consists of two distinct membranes. The outer membrane is continuous with the endoplasmic reticulum, connecting to the cell's protein synthesis machinery. The inner membrane lines the nucleoplasm and nuclear contents, providing direct contact with the genetic material inside. Between the outer and inner nuclear membranes lies the perinuclear space. This gap is remarkably narrow, measuring only 20 to 40 nanometers wide. Despite its small size, this space plays important roles in protein modification and cellular signaling. Nuclear pore complexes are large protein structures that perforate the nuclear envelope at sites where the inner and outer membranes fuse together. Each nucleus contains thousands of these pores, creating regulated gateways for molecular transport between the nucleus and cytoplasm. The nuclear envelope serves multiple critical functions. It protects genetic material from potential cytoplasmic damage. It controls the passage of molecules in and out of the nucleus, ensuring only appropriate substances cross the barrier. It maintains a distinct nuclear environment with different conditions than the cytoplasm. Finally, it anchors chromatin to the nuclear periphery, helping organize the genetic material spatially. Beneath the inner nuclear membrane lies the nuclear lamina, a network of intermediate filament proteins. This protein meshwork provides mechanical support to the entire nuclear envelope, giving it structural stability. The lamina also plays a key role in organizing chromatin, positioning different regions of DNA within the nucleus. The nuclear envelope is a dynamic structure, not a permanent fixture. During mitosis, the nuclear envelope disassembles to allow chromosome segregation. Chromosomes need direct access to the mitotic spindle for proper division. After division completes, the nuclear envelope reassembles around the two sets of daughter chromosomes, reforming the nucleus in each new cell.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.